Welcome back to Flagship FutureCast. Our guest today with our founder and CEO, Noubar Afeyan, is Aviv Regev. Aviv is the head of early stage research and development at Genentech, as well as the executive vice president. She is also the co-founder of the Human Cell Atlas and a powerhouse woman in science behind the Broad Institute and many scientific innovations we'll hear about shortly. So with that, Noubar and Aviv, thank you.
You're here. All right, well, welcome. Thanks for making time for us, and otherwise, I'm sure, a very busy week, although at least we're dry in here. So we've got a good setup. So Aviv, let's just dive into it. One of the areas that I know you've been active in for quite a while is the intersection between data science and biology, and more recently, with AI and machine learning becoming kind of even more useful there. I guess it would be good to start out with getting your thoughts on what is maybe unique about the biology applications of machine learning and AI that the audience should be aware of, and how do you think about the convergence of those spaces?
Yeah, I think it's a terrific question. Maybe I'll start by saying that I think when you think of a tool and you think of a problem, you first want to figure out whether the tool would actually help what's material about the problem. And in biology, I think that in biology, especially in medicine, drug discovery and development, the biggest things that are stumbling blocks for us today are exactly the things in which computational approaches, and especially the modern approaches in kind of the current incarnation of machine learning, tend to show most of their strengths. So first of all, we need to make a lot of predictions. And historically, we don't make them so well, meaning we predict, and most of the time we're wrong. Mm-hmm. Good or bad, we just end up being wrong. And so being more predictive is something that we need to be. Second, our processes are processes of making new things in the world. They're not just about figuring out how biology works, but if you want to make a medicine is a new thing that didn't exist before. So we need to generate it. Mm-hmm. And so you want things that are generative. It can imagine new things for you. And that's another strength of machine learning. And the third is just the sheer scale of biology is the kind that humans find difficult to hold in their hand and kind of see the forest for the trees, see the general pattern in a lot of details, and that's a representation problem. And that's, again, something that machine learning is particularly strong at. So there's something there that suggests that the marriage should be good, because this party has a lot of problems, and this party has things that in other domains have shown themselves to be successful. They can make amazing predictions. They can generate new things. I mean, people now see all the art and text and stuff like that, even what we think of as creative things. And then they also can deal with the space that is so big and show the forest for the trees, find the general pattern. Now, there's also ways in which biology is very, very special. So I'll enumerate some of it. I already mentioned its size. The size itself is not really why it's so bad. The thing that really makes it bad is that it's nonlinear, meaning the whole is different than the sum of the parts, sometimes bigger, sometimes smaller. If I look at what two things do, say, two molecules, two interventions, two drugs, I can't really predict what their combined outcome would be. If we could, our lives would have been a lot easier. And historically, that's been a big stumbling block in biology and in medicine. If you want to figure out how a cell works, you perturb a gene, you see what happens. You perturb another gene, you see what happens. You need to perturb both of them to see what would happen if you manipulated two. You can't just sum up the two effects. And because there's many genes, this explodes combinatorially. And combinatorial explosion means we can't actually do a systematic test of everything that's out there in the world. That's a big problem in biology, and it's a particularly interesting problem for computer scientists. It's what we call causal inference, to try to understand the causes of things. And machine learning has proven that it does a particularly good job on nonlinear systems that before were actually very difficult for computer scientists, not just for biologists.
So on that thought, I guess, so biology is a field in which human understanding, of course, is rate limiting, and we kind of try to do things in a way that maximizes our ability to understand and therefore predict. Then you have now machine learning tools, which also humans don't understand how they do what they do. So now we have two systems that we don't understand that are interacting and trying to help us make progress. I'm not asking the trivial side of this question, but then what of the humans? In other words, to what extent do we need to insist that the only things worth working on and things that humans can not only understand but write a paper around, when in fact neither side lends itself easily to that? And frankly, neither side really cares about whether the human understands it.
I love the way that you put it, first of all, because one of the things that I think wouldn't be an ideal solution, you could end up with some god-awful huge model of all of biology, and essentially you have that model already. Imagine that was a cell. Well, I can grow a cell in a dish now and perturb it, manipulate it, and see what happens. If all I end up with is basically, in a sense, a replica of this, and I don't know how it operates, then in the end, all I'm going to end up doing is more experiments, but maybe I'll do them in silico instead of in the lab. Well, in silico might be cheaper, easier, faster, but do I actually understand what is going on? There is a version of that, and we have to be careful around it. So here is where the human element is absolutely critical. There's two crucial steps for the human biologists. By the way, humans also write the algorithms. They don't write themselves, at least not yet. It's only a matter of time, maybe.
Soon. Maybe today.
But, well, we already do it for matrix factorization.
Exactly,
That's amazing.
exactly.
Well, first of all, you have to formulate a question.
Yeah.
And the question has to be important. There's endless problems to solve in the world. There's endless theorems to prove and things to predict, but most of them don't matter at all.
Yeah.
And knowing what's a good problem and formulating it in a way that's meaningful, that requires humans, at least today.
Yeah.
Then you touched on the other moment that's absolutely crucial, interpretation. It is still true that with all the fancy machine learning that we apply to beautiful biological data, after all that is said and done, a human still shows up and tries to understand what's, again, interesting and important there. And we still don't know, at least to date, to train algorithms to tell that for us. Hopefully we would one day. Would only make us even better and stronger and faster.
Yeah.
But those are crucial human elements, even just in the computational endeavor. However, I want to say one more thing on that. The way we talked about it until now was like, here, computer science has these great things to give, and biology really needs them. I actually see this as much more reciprocal. Biology has a lot to give. Biology as a field, or medicine as a field, has a ton to give, or chemistry as a field. Besides defining the right problems and seeing where the meaningful answer is, they have data that needs to be generated in an imaginative way and not just brute force, because brute force will never solve the problem. It's too big for all experiments, more than atoms in the universe, so definitely too big. And in biology in particular, we have the ability to do interventions. Either we do a clinical trial, that's quite a magnificent interventions, or we're in the lab and we're using genetics. In both of these cases, we can actually intervene in the causes. That is something that most other fields in the world cannot do. When somebody does A/B testing in tech, they don't actually go into the user's brain and pluck out a neuron and ask, well, I wonder what would happen now. But in the biology lab, we do things like this every day, not to humans, but to animal models, to cells. And so we get real data of a different kind that exists in other fields. So our problems are different, our tools are different, our thinking is different, and it's going to change computer science.
Yeah, maybe I'll just add to that, at least the way we're working on it and thinking about it, is that in the generative side, which you're describing, when computers are generating novelty, which we also don't understand why they did that, in a way, the experience of biology, which is another system we don't understand, might be instructive of how computer scientists could understand what their generative algorithms did, in that they're both black boxes.
Absolutely.
So the same approach to perturbation, like if you don't know how a game's being played, one way to study is to essentially play the game with one of the steps changed and see how it changes, which is kind of what we do when we perturb a gene. So there may be like techniques we've used as humans to try to understand as best we can. So related to that, you also mentioned about data. So we've generated data, as much of it as we can historically, and with hoping that we can get the statistical approaches to try to harness it. Now we have better tools. How should the data generation change to deal with the new reality?
Yeah. So there are many dramatic ways that I think data generation should change. I'll try and enumerate as many of them as I can remember. The first one is what we like to call the lab in a loop. Actually, the biology, the experimental biology, or the experimental chemistry, and the computational work should not be seen as two separate things. They're actually tied at the hip. You need to do different kinds of experiments because of the computation you want to do, and you need to do different kinds of computation because of the experiments that you can do, and you want to iterate fast and furious. If I'm trying, for example, to generate the next antibody in a machine learning, at least assisted way, using generative models, or to do the same for the next small molecule, or to figure out the mechanism of, how should I manipulate my cell therapy, any of these things is going to fall or generate a better personalized neoantigen vaccine, all things that are near and dear to both of our hearts. In each of these cases, I might need to do experiments differently because of the algorithm that I use. So I'll use the antibody example. I will do experiments that if I'm just doing the traditional antibody engineering, I wouldn't do. They're not going to serve me immediately for that purpose. But if I'm using, say, a reinforcement learning approach, I need to generate examples that would reduce the uncertainty of the algorithm about its own predictions.
Right.
So I'm going to go and synthesize molecules, make antibodies, do many different things that I wouldn't do otherwise, just in order to make my algorithm generally better. And once it's generally better, of course, it's going to generate better molecules. This all touches into what I think is at the heart of creativity, human and algorithmic creativity. And this is generalizing out of the distribution, what we call out of distribution generalization, or OOD generalization. We have all, humans and algorithms alike, have seen some things in the world. When we think about ourselves as imaginative, a great medicinal chemist, a great biologist, it's this stroke of kind of thinking beyond where you are. The same is true for the algorithm, getting beyond, far beyond where it trained itself based on things that it's already seen. If you can do that, you're extremely powerful. And most of the time, algorithms and humans struggle with that.
Yeah.
That ability to kind of jump beyond. And what we need to generate in terms of data is what allows us to do that, either to the human or to the algorithm. I also think we need to change how we generate data, not just the scale, which is extremely important, but appreciating that as you go beyond a certain scale, you can let go of some things that sometimes we held on to very, very tightly. For example, people always think the data needs to be perfect. The maximum that I can have in a particular experiment or a particular measurement. Well, single-cell RNA-seq is not like that. It's actually terrible. It's noisy and messy and so on, and at first people didn't like that. But it turns out that it's better to have lower quality data per experiment and more experiments, or in this case, more cells, than higher quality data, but fewer cells. And that's because the algorithms can do better with the scale, and they clean up the noise once they have enough scale. So learning what we can let go of and what we get in return, what's the trade-off between those two things, is very important. There's things that are better done randomly. That's how the immune system finds antibodies. It's a search algorithm run by molecules and cells inside our body every day. It does a random search. It introduces all sorts of random mutations, or semi-random, only in certain places, right? And the same is true for T cells, for BCRs, and so on. And then it tests them, basically. It's called a genetic algorithm. And it finds amazing things in this way. When we go and engineer antibodies, not just me personally, but the world at large, that's not what we actually do. We actually go and do very fine things based on our depth of knowledge and our understanding. And that was very sensible when all you could do was 1,000 things at a time, or 100 things at a time. But what if you can do 1,000,000 things at a time, or 1,000,000,000 things at a time? All of a sudden, the approach that just goes at random is sometimes better than the approach that's very well designed.
Well, you used the expression, it's a very sensible thing to do, which basically is an entirely human concept, sensible. And it's a very human thing to do. Well, I know, it's a very human thing to do. I agree with that. But so that kind of touches on something that I think it would be worth just expanding a little bit more, which is you talked in a few ways about both humans and algorithms wanting to or attempting to go beyond what's adjacent and what's kind of seemingly reasonable, and whether algorithms could do that. And it kind of raises this topic that we were talking a little earlier about the nature of risk as opposed to uncertainty. One of the things that I think it's beyond simply the fact that uncertainty, these are two different things, but also humans, it's been well documented, have a preference for risk, however low it is, over uncertainty. And moreover, they basically assume that uncertain things are inherently extremely high risk. Even though, if you just kind of look at it, there should be low risk parts of uncertainty and high risk parts of uncertainty. So and I know that science is about kind of taming these uncertainties and moving them into the column of certainty with knowledge, but yet humans desperately resist that, even scientists.
Yeah.
So how to think about these two natures? Should people run away from uncertainty into the comforts of known high risk, which is drug development, or early stage drug development, or should they instead look for low risk parts of uncertainty? And then how do you actually justify getting budget money to work on uncertain things?
Yeah. Oh, that's a big question. Let me
Well, you have a big
let
job.
me start. Let me start with the very end of what you said. How do you actually get to the resources to do that? And I want to say that's one of the aspects in which I'm really fortunate. I think I'm in a place where people believe in it, that you actually have to hit at the uncertainty problem, and that you have to be willing to commit to it, and you have to suspend your disbelief. It helps that there is a track record of success for the approach, because the more something has been successful, the more people consider it lower risk, even if it's actually extremely risky. And that's I think, a cultural thing all in its own right. So I don't have a great answer on how I personally get people to invest into it, because I did come into an ecosystem where this was a highly held value, and I probably wouldn't have come otherwise. Mm-hmm. It's also a highly held value in my own mind. Then I'm going to go and unpack some of the other things that you said. I'll start with the fact, tying it a little bit to what you asked me about the experiments. Running these kinds of experiments is one of the ways that you get around this human bias. Running large screens, for example, is one of the ways that you take people, you peel people away from the genes that they like, right? Every biologist you will ask has genes that they like. I do too, by the way. You get very attached to them over time. It's your knowledge, it's your thinking, it's just stuff that speaks to you, whatever. You discovered them, that's a big one that makes people like certain things. Screens that are comprehensive, I never use the word unbiased, and I teach all my students never to use it because everything has biases, but screens are comprehensive, and you often don't make those kinds of choices anymore. So they let you kind of march into the uncertain part of the world without really realizing even necessarily that you're doing it. It's maybe conniving a little bit, but I think it's extremely impactful, and it's something that you get trained into, you get used to thinking in this way. If we had actually applied our biases all the time, it would be really negative for us. And that's one of the ways to get out of that, and that's why I love randomness. Throwing in some randomness really helps you, both in your algorithms and in your very practical application of tools and methods and things like that. People love talking about, the 20% time that many companies allow employees to use. That's an example of that. You have no idea what they're doing with it. You have no control over it. You do not review it. It is completely uncertain, and it's a great way of just being there without trying to impose all our wisdom, which is probably not as wise as we would want it to be. So that's one, I think, aspect of taking it on. The second aspect of taking it on is, I think, interdisciplinary interactions. Because one person's uncertainty is not another person's. And by bringing, especially one field's is not another field's. The things that feel very certain for one field are often the things that feel extremely uncertain for another. They're so uncertain they don't want to think about them at all. They make them very uncomfortable. By bringing people like those together, you have a huge opportunity to unlock that part, and actually to see very interesting challenges happen by humans to other humans in ways that I don't know other things that create that kind of magic. And that's that R&D style of magic that people always aspire to. You have to bring different people together to make that happen. And I think increasingly, human and algorithm serve this purpose for each other. That's, again, in that moment of interpretation or problem formulation or looking at the results, a human can have huge implications in saying, this is where it's doing something, this is where it's not, is one of those things that could make the algorithm go in different ways, just like when somebody does it to us. But the algorithm itself is another entity in that conversation, and it can help the scientist go to those uncertain places that otherwise it won't.
So you're saying algorithms need humans to really
do well? Oh, yeah.
Very
much so.
Related.
Currently, they're mostly encoding humans.
Yeah,
They're
exactly.
actually encoding. They're encoding our own language. I don't mean encoding in the technical sense, although some of them also encode in the technical sense.
Yeah.
but they are learning to imitate us.
Digital twins, yeah.
To imitate us.
Yeah.
And that's fantastic, by the way, because especially in medicine, if we had something that imitated humans well and was not humans, that would solve a lot of our biggest challenges in development. We would love to do that. We would love to have human biology without the cost to humans.
Yeah.
So, of course.
Yeah. So, in fact, it's one of the things you said in passing. I just want to touch on is just to comment on is that, you know, in a way you said, you know, if you introduce some randomness, it kind of overcomes some of the favoritism or biases that we have. You know, when we think a lot about making leaps. So, kind of like what leads to leaps. One of the interesting things that comes out of what you said is that randomness in a way destroys adjacency. And we tend to get comfort in working on adjacencies so that one thing leads to the next, leads to the next. But if you introduce randomness into that, you lose that sense of adjacency, which allows for leaps, because you kind of make unexpected leaps because you lose the comforts of incremental steps that are based on everything else that you thought you knew. So it forces leaps in a way. So introducing randomness may force leaps to be possible that otherwise would have taken a lot longer. So it's a good way to think.
And that's why you want scaled methods, because with scaled methods you can afford to do that. You can only afford a certain amount of randomness in your life, right? The 20% time is a certain thing that you can afford. The amount of other things that would never yield that you still do. But the cheaper each of them is, based on any economic calculations, it can be in dollars and it can be in people time and in any other thing. The cheaper it is, the more you can afford it. And then that is what scale actually gives you. It gives you the ability to do that. And once it does, it unleashes things that you, honestly, none of us can imagine.
It's interesting. I mean, it's not kind of where I was going to go, but I'll ask you anyway, because you've straddled the academic world, you've straddled the industry world now as a scientist leader. You know, one of the things that you used the word emergence before, nonlinear kind of emergent things. So one of the things I would observe about, you know, people, you know, kind of even science in general, wherever it's done, is that as insights emerge, as there's major, you know, gains of function, if you will, in our understanding, the people who are there when that happens essentially describe how it happened in ways that are highly non-emergent. I've never seen a paper written in a truly emergent way that says this accident followed by this meeting followed by...
The paper would get rejected.
Well, that's right. So you kind of have to almost reverse engineer a rationale that is linear, is sufficient for people to understand it, but unfortunately it leads people to further believe that a set of linear steps could lead to unreasonable advances. You're saying we're going to do much more of this kind of thing. So can you imagine a paper that results from a very large, you know, screen data, big data methods that lead to something that really has nothing to do with what came before? What would you reference, and how would you explain it sufficient to publish it? Is that an impediment?
It is, and I can give you real-life examples of how bad the impediment can be. So since you were reflecting on my older life, so I'll use an example from that older life. We actually had a study like that with random promoter sequences to try and develop a model that, given any sequence of a certain length, because that was the training data, would be able to predict expression correctly. It was done in the East. There's no reason not to do it in the human, but it was done in the East. One of the cheapest experiments my lab has ever run. It didn't take that long to actually run the experiment, to build up the experimental model, to build up a model, a computational model from it, write a paper. But it took 3 years to publish the paper because people really felt uncomfortable about it. And by the way, that's a very common pattern. You said when a new finding emerges, when a new insight, and it can be a technical insight, it can be a biological one, there's usually actually a lag time between the moment that happened and the moment that it really took off. And that lag time is people getting used to it. And by the way, those who get used to it early and realize it happened 3 years or 2 years, that's the arbitrage, right? You can often see their careers take off if it's in academic science, or they would make, you know, a great commercial success if it is in a more for-profit world. There's many different ways in which it will come to them, but often it's because they actually understood it earlier than other people. They didn't make the discovery themselves. They just realized it was there. That lag is, again, very human. It still feels uncertain to somebody when it actually became certain, but they haven't realized it yet. And in fact, they're trying to convince themselves that it hasn't happened. How many times have you heard people say, no, it's not such a big deal. Or it's not really different. Or it's not this, or it's not that. Because it's like, yeah, that's kind of human nature. It's the homeostasis of our brains
Yeah.
to think like in a pre-adapted way rather than about the future. So definitely these things take their time to kind of sink in and become real, and they're definitely difficult to put out there in the world, and that includes the world of publishing.
Let's talk about talent a bit, right? So we're describing, on the one hand, a continuum of better and better abilities to do science and biology, aided by computation, et cetera. But also, enough. The continuum has reached a place where it might affect the kind of talent and mindsets you need. So, as you've gone from academia into industry, both, in my view, do equally cutting-edge science, at least my experience. How do you think about the talent side of this? What kind of mindset, scientific talent, do you find can handle these different dualities and uncertainties?
Yeah, so first of all, I would say, and I said that earlier too, I think you absolutely need to have multiple mindsets. Mm-hmm. You don't want just one. That tackles a lot of the challenges that we talked about. But in particular, and I'm going to use here, I think, an analogy for myself, you want people to be able to speak with an accent. So I was not born and raised here. I speak with an Israeli accent. Noubar also has one, not Israeli, different one. And I think one of the things that I realized over the years is that I actually make less and less of an effort to not speak with an accent. I don't care about it anymore. I want people to understand me, but that is it. The rest of it, that's who I am, but it's also very liberating. I can speak everywhere about anything. I don't actually cycle too many neurons on the phonemes and things like that. What I sometimes feel holds people back in the science, now in the analogy, is that if they are not native to something, they are so embarrassed about their accent, about not being the expert, that they'd rather not talk at all. They just stay out. And that is a huge loss for all of us. So one of the essential things with talent and with bringing people together is bringing people who are different and have different skills, but letting them speak with their accent in the other domain. Letting the computer scientist actually have real life opinions and stated views and impact, say, in biology or in chemistry or in whatever. Letting the chemist become a biological expert, letting the biologist become a computational one, and so on and so forth. That is one of the critical things. And that, by the way, is not just in the Genentech. That was true at the Broad, too. And I think it's a big barrier, actually, for our fields unless we get over it. Because medicines will only get made by multiple people who are very different from each other working together. There is absolutely no other way to make real medicines that make it to real patients and have a real impact on human lives. You can do a bit here and a bit there, but you can't do all of these bits together. So this speaking with an accent is going to be something that's going to remain. Now, there are going to be kids, like my own son, who happen to be very bilingual. They somehow grew up in this magical situation where they speak two extremely different languages from each other, and they're both fluent in all of them and everything like that. But that shouldn't be the bar. That's kind of, yeah, occasionally it happens. It's awesome. When somebody like that comes in, absolutely, we love them. But we don't have to have that. It's totally fine. You can learn a language. You can learn it at any point in your life. You don't have to learn it in the crucial first 8 years when you learn it as a native tongue. If you learn it later, it's fine. We're smart. We learn. And we just figure it out. So that, to me, is really the essential piece around the talent. Now, there are specific things that come to play in this. We're a very established place. We existed for a long time. We're very proud of that. We've done a lot of things during these years. We have people who are unbelievable in their smartness and expertise. And I agree with you 100%. There is innovation everywhere. And it is as cutting edge. No one got like a special, IP protection around. They have cutting edge and the other people don't get to do it. Everyone does. But when you want to move to the next opportunity and the next opportunity that will come after that, and today we talk about machine learning, and we could have talked about other opportunities too. And in 5 years, we'll talk about something else, I'm sure. You have to have everyone actually feel that they're equals in this. The people who are there, the new people who are coming in, they might come with very different skills, but they don't know this language. And the people who are inside, they might not know this other language. Everyone is learning something. And everyone is switching their thinking along the way. And what you always want to hope is that, 3-4 years down the road, people forget. They don't even realize they didn't know something before. They don't even realize this was not native for them a few years ago. That's who they are right now. right? Like I am an Israeli American now. I was only an Israeli 20 years ago.
Well, very interesting. You're touching on a bunch of different interesting themes. You know, I think this also applies to lots of immigrants, you know, who kind of bring these accents as well. It's interesting, though, kind of the way in which emergence, which we talked about, and expertise might actually be concepts that one has to really think about when they come together, because expertise is better when there isn't emergence. If things are this, you know, predictably sum total of things and you have expertise, then you know, kind of you set the accent.
Yeah.
But within wave after wave of emergence, to some extent that expertise is short-lived.
Yeah.
And being a dynamic expert as opposed to a static expert isn't for everybody. I mean, people work hard to become viewed as an expert, and then they cash it in the way they're treated and kind of worshipped, as opposed to saying, no, actually, it's a transient expertise because things are emergently going to change. And of course, these algorithms are going to force that at some level. So what you thought, you know, a month ago, 2 months ago, how you designed the protein is different than how you design it today. Imagine how experts feel of that, right? So kind of, either you kind of have to just keep swimming, or so it's an interesting kind of talent application.
I still have a huge, huge respect and regard for expertise.
Well, sure.
Because there's kind of the knowledge base that goes along with it. There's the associative learning, so finding the connections in it. There's the classifier that gets trained in your head to really know when something is off, which is a huge strength of experts and saves a lot of wasted time and effort. But also because the real masterful experts, they have honed their way of thinking. It's not just about what they've seen before and what they know and what they love and so on. They kind of they can also seize with their expertise into a new problem where it's truly uncharted territory. And they can, if there's one thing they gained, is that they gained the trust in themselves that even though they have no idea what they're doing, they're still going to figure something out. And that is huge in drug discovery and development, huge, because you're going to have moment after moment of having no idea what you're doing, because no one knows. It hasn't happened yet, right? That's big, and it's big in scientific research too. So I have very, very deep respect for the experts. The place where you want to inject this randomness is around everyone, self-included, has their way of doing things and their way of thinking, and you hone it and you hone it. You get really great at it. You don't want that to become autopilot. And when new things come, often people first start using them in old ways. Now, again, I've seen that with single-cell RNA-seq. It's a great example for that once it became really a generally used tool, people tried to shoehorn it into the way that they would have done things in their old lives. It's just kind of a better way of doing the old thing. But it actually opened up things that were entirely new, and that was the best stuff to do with it. And that takes time. And that's where expertise, I think, is sometimes in a little bit of tension. Not that you want to resolve it completely, but expertise can be in a tension with innovation sometimes.
Yeah, I think what you're describing as the best of expertise, at least in my experience, is the limited set of experts who are actually dynamic and who are regenerative experts, as opposed to assertive experts. But that's a different
expertise.
Well, I mean,
that's beautiful.
I think that's key. You almost have to be doubtful about your own expertise in order to stay an expert, is the way you're describing it. Whereas if you, so anyway, just one, maybe I guess we'll end at one thing. So you've been involved in the, obviously led a lot of the efforts of the Human Cell Atlas. We did the Human Genome Project before, and we're getting better and better at creating the kind of the list of parts and the way in which they may interact, etc. Where do you think we are as you look in the future about what becomes possible with this that might represent big step change leaps? It doesn't just have to be with respect to the application in drugs, but in general, what is becoming possible as these pieces come into focus?
Yeah, we actually, Sarah Teichmann and I just wrote a piece about that. So not that it's a plug, but it's front and center in my mind. And I think one thing that actually struck us is that we're living more in the future that we wanted to imagine than we expected to be maybe at this point. So Human Cell Atlas, or HCA as we like to call it, moved kind of faster maybe than we expected, or the whole community did. There's more cells, people have done more tissues. The algorithms are better. I think the biology understanding is deeper. So it's kind of nice to see it also play out in action in front of our eyes, but there's still stuff to kind of look forward to much more into the future. I think the first level, you have kind of a basic utility level that just takes out the uncertainty out of many things that used to be uncertain, and you just have your reference map, and you just go there, and you get the answer. And it's almost like boring. It's great when stuff becomes boring. It means it has been commoditized completely, and you just do it every day, and you forget that it used to be difficult. And I will start with the drug discovery example. I mean, we target molecules, right, in the body. We want to know where they're at because we want to avoid adverse events later on in toxicities, on-target toxicities. If you don't know where the gene is expressed, that's actually a lot harder. If all you have to do is go into your nice model that already integrated over 100 million cells from about 10,000 individuals, these are all real numbers, from about 10,000 individuals and covers about 18 organs and systems, which is the vast majority of the body, it's like 2 minutes away from you, the answer. And you're done. It's not perfect. It's still based on RNA. Yes, the proteins will come. Yes, there will be also in situ. These are all the better versions of the same idea. But it's like that. You shouldn't anymore think, oh, I'll get to that later on when I do my safety assessment years later. I can ask this question today before you even decided to embark on the target. And there could be things like this, and there could be things like that, and you would have to apply a lot of judgment. But those simple things, they're a big part of it. Or you're designing a cell therapy or a gene therapy that you want to target the right cell. Well, if you actually know the cell, that's going to help. Or you need an enhancer for your AAV. Well, if I already have all the enhancers in all of the subtypes of cells, wouldn't that be helpful? These are going to be boring things. There's not going to be anything interesting about them. And not in the distant future. And they shouldn't be interesting because they're just a first barrier that we've had. And they were difficult before, and now they will be easy. So that's the first one. If I dig a layer deeper, I think the questions that I find particularly fascinating in this is whether we would end up with a pretty full vocabulary of what are the programs that the body runs. Because again, taking the medicine perspective on it, there's plenty of examples in the current pharmacopoeia of drugs that were designed around one thing and then turned out to also be great for this and to be great for that and to be great for this other thing and that other thing because the program, the process actually that's impacted by this drug is active in a lot of different places. And now not in the negative sense, but actually in the positive sense that it's important. Inflammatory processes tend to be like that. Well, having a comprehensive vocabulary like this, maybe not everything, but a really good sampling of it would be amazing because we would actually know what we're working on. Moreover, we would realize all the targets that are actually part of the same story, the same program. So we don't actually should look at each of them individually. Maybe one would be enough. We're increasingly learning that combinatorial effects are mostly limited without modules, which actually takes that combinatorial problem I talked about with the nonlinearity and reduces it orders of magnitude away from where we thought it might be. But that means that if we are looking for synergies, we probably actually should look for things that are adjacent to each other. And if we're looking for additivities, then things that are far away from each other. It's a crude estimate based on very little data, but that's the kind of stuff that we're starting to learn from these kinds of pieces of information. Don't run and do your drug discovery based on this. It's several years away, right? But several years is nothing. We'll be here before we know it. So that's a second maybe layer of understanding that we would want to have. And then one after that, and the one where I think really the full glory will come from is tissue biology. We still very much think in genes and cells, but we should be thinking in tissues. Multiple cells of different kinds interacting and affecting each other. That's the heart of homeostasis, which is what we want to restore by therapies, which is, what gave the great success of immunotherapy in cancer. That's a tissue biology question. It's one cell type and another cell type interacting with each other. And now we realize in 300 other cell types of their best friends. That kind of understanding is possible only if you have the units. The units are the cells, but it is much better if you have them spatially organized and understand all of their interactions. It's what we call the spatial branch of the Human Cell Atlas. And that will come again. And I think many, many things will kind of fork out, especially from the technologies that HCA really made robust. So for example, the ability to screen cellular phenotypes using full molecular profiles at scale that can be applied to a small molecule scale doesn't sound like insanity anymore. And it will bring things like phenotypic screens and mechanistic screens a lot closer to each other than they used to be. People used to think of them as two completely different things, but there's actually only one cell, and it's only made of molecules. There's not ether involved, And if we could actually screen in this way, then we would get answers a lot faster and at a much greater depth than we've had before. We're not fully there yet, right, as a community. It's going to happen.
So this answer you just gave is interesting in the context that this notion of tissues and getting closer to the human. So in one important way in which going from the broad to Genentech, I guess, must have changed your scientific purview, is that what you're working on ultimately is now going into humans. And the science of going into humans is a fairly kind of underdeveloped science, although there's a lot of advances being made, in that we can do a lot in labs, but in humans it's kind of fairly limited what you can do. We're getting lots of data, et cetera. Now, we're just coming out of a pandemic period where drug development was done or vaccine development was done in a fundamentally different way than anything else has ever been developed, and yet produced an immunological effect that has been helpful. How do you think as a scientist leader about this period of why aren't we going to do similar things for other diseases? And is this a time where the added data, other computational added models should give us confidence that it's not such a black box so that we're forever going to have to say, well, we got to do 5, 10 years of this before we can actually do that. So how do you think about the scientific approach? Talk about being an immigrant. You're kind of an immigrant bringing science into something that's much more kind of human oriented. How do you think about all that?
Yeah, so starting with your immigrant point, one of the things I learned as an immigrant is that once you immigrate and kind of realize another place, another country, you realize, in country A, people do things like this, like they put the stamp on one side of the envelope, and in country B, they put the stamp on the other side of the envelope, and none of this matters. You could have put it you could put it at the bottom and not at the top. You could have put it on the back and not on the front. I mean, it doesn't actually matter. And what it means is, first of all, you have option A and option B, but you actually probably have C, D, E, and F. You just never thought like that because you were living in your own fixed set of assumptions. So to me, what you've just said about the pandemic is similar to that. We lived in another country for a while. We saw a different way of doing things. And we should take a step back and say, well, in that case, maybe there's that plus all sorts of other ways. It's liberating. So yes, I definitely moved to Genentech because of a one-word explanation, patience. I really wanted to see that happen. And so I decided at some point to pack myself up and the family and so on and go to another coast and try and give it a whirl, but with all seriousness. But at the same time, if you think about it from putting patients first, then you can't afford not to think about maybe there are other ways. At the same time, it's not just being realistic, it's understanding the differences between things. What could work for an infectious disease with a huge patient base, unfortunately, because of a raging pandemic, might not be the same timescales of disease even for a common complex chronic disease that takes years or decades to develop in the body. It doesn't take 10 days. It's a long disease. So, of course, the timelines of clinical development are going to change for the mere fact that you actually have to wait longer to see effects for certain things. That's understandable, that the statistics of the phenomenon are different. The size of the trial could change. We have to combine, not balance, but combine these two worldviews. Many things will change. There's a religious, Talmudic saying, Justine will appreciate. Prophecy was given to the fools. So you have to be very careful when you prophesize about the future. Nevua nitna leshoftim. That's in Hebrew. But at the same time, if I think about it from a science point of view, I think if we reached a world which was more programmable for biology, then we would have many, many of the benefits that we've seen in the pandemic. In the pandemic, to some extent, at least for the vaccines, and you know it better than I do, at least for the vaccines, there was a high component of programming the medicine. Mm-hmm. The medicine was programmable because we understood the virus. So we had the genomics of the virus. We had the protein structure. There was beautiful structural biology involved based on decades, actually, of work in structural biology. It didn't just appear by magic. You and I both know none of those things appeared by magic. In a year's time, they were based on decades, actually, of work and development. And then you kind of had the more programmable side of biology that we understand better, where the epitope is, what you would want to actually vaccinate with, and so on. And then on the other end, you had an actual programmable entity, which was an RNA molecule. Right. And that was the perfect setup in a sense. All of these components together. And so now you want to try and work from that and extrapolate, well, first, other kinds of vaccines, cancer vaccines for neoantigens in patients, programmable. Machine learning is extremely crucial there because you actually have to decide what are the antigens to vaccinate against. And that's based on a model that you can then personalize for different individuals. Again, you and I both know that from the perspective of different programs. Cell therapies, making cells programmable. So it's not just, first you program them just to target the antigen you want to go after, but you also want to program them to have all sorts of great properties inside the body. That's something we work on. All of these perturbation screens that we talked about and things like that, that feeds directly to that. Great. But maybe these other therapies that were more traditional can also be programmable. Antibodies, well, the body programs them. We just have to figure out how to search as well. Small molecules, things that we don't think about like this today, maybe they could get closer to that place. So these are many, many layers from a pure science technology perspective that you can think about bringing some of those benefits. And then, of course, there's the process.
I'm going to end on one question since you went to the Talmudic route and see.
And I might need help from Justine. I'm not that great.
No, but I'm going to see if you go there because it's something that I've been thinking a lot about of late. So, in science, you believe based on facts. In other fields, faith is belief without facts.
Yeah.
So when you're dealing with uncertainty, I'm going to posit the case and see how you think about it. You'll have your own reflections. If you deal with uncertainty, you could, based on science, not believe because there's no facts. But unless you're willing to believe without the facts for a little while, which is otherwise faith, something discouraged in science, you'll never do the experiment needed to figure out whether the belief was justified or not. So how do you toggle between what is something that people stay away from, which is belief without facts, with something that eventually becomes fact that derived from your willingness to believe without the facts?
That is what we call the testable hypothesis in science. You're allowed to hypothesize to your heart's desire. That's the belief that you want to hypothesize. You actually have to come up with an hypothesis. And it is testable, and you need to run the test. You can't pre-answer it before you have the answer, but you also shouldn't pre-answer it with, I'm not even going to do this because I don't believe it. Especially not where patients are involved. And those are hard decisions to make. If there's kind of the one thing that I would highlight as, for me personally, my distinguishing moment of moving from the two worlds, it came fast and furious. And that was making these hard decisions in the face of uncertainty that people do every day. I was actually humbled by it. I was like, look at these people. Because in academic science, the stakes of the decision feel different. Here, they're very, very high, but you still have to make them. And the decision that you make is actually to test your hypothesis, to gather the facts so that you will actually test what you believe. You can't just go on belief. That kills you.
Sure.
But you have to test, and the test is hard.
So related to that, maybe just a little fine point to that, in academic science where the goal is to advance knowledge and publish, I suppose you're better off being ambivalent as to whether the hypothesis is or the experiment works or not because you're doing the experiment. But when it comes to patients, are you as a scientist able to be optimistic that your hypothesis may be true until the experiment is run and it is or it isn't? In other words, how do you deal with the emotional side of this?
Yeah, I think realizing that in every waterfall plot or any other way you plot your data, every bar is a person. That person has a family. That person, their lives are sometimes in a balance. Definitely their well-being, and in many cases, especially in oncology, truly their lives, is something that haunts you, but in a positive way, in a sense, every single day. And you have to take those decisions with the utmost responsibility. But you also understand at the same time, you do everything that you can preclinically, and at some point you will only get the answer after. And kind of to come full circle to what we started with, this is why it's such a passion to make biology more predictable. It's my personal passion too. If you actually make it more predictable, it would reduce the uncertainty. And if it reduces the uncertainty, that is our responsibility to patients. And at the same time, you bring the best science that you have to bear, and yet you're humble at the same time to know that you don't really know.
Right.
And you're very, very grateful to the patients and to their families for being willing to try it out with you. Because that's what we do every day.
That's a great place to end. Thanks so much for your time.
Thank you.
Great conversation. Thank you. Thank you.